Welcome everyone to my channel BioWorld once again. So today with the chapter Absorption by Roots, the next process after uh, innovation and diffusion, that is osmosis. Now osmosis is one of the most important process here, and how the water molecule passes from the soil to the root, and what is the process of phenomenon osmosis is, we come across in this video today. So osmosis is the movement of water molecule. Please do notice the keyword. So water molecule is one of the keyword. So it is a movement of water molecules from the region of there. Remember, there. That means it is talking about the water. So from their region of higher concentration to their region of lower concentration. Remember, it is talking about the concentration of water, not the solute. And the written here, the words, please do mention it, dilute solution. Dilute means the solution which has more water and less solute. That is more solvent and less solute. And concentrated means what? Concentrated means when the solute is more and solvent is less in terms of chemistry. So dilute solution here, it is talking about the higher concentration of water. So the movement is always from the region of higher concentration of water, where water is more, to the region of lower concentration of water. Remember this. Concentrated here does not means or implies to the solute concentration. Here it implies to the concentration of water molecules. So remember, in soil the water is always high, that is more in compared to the cell sap in the root cell. So the movement is always from the soil into the root. And what is the next keyword? Or with a higher solute concentration through a semi-permeable membrane. So this third is the most important keyword. If you are not writing in a definition semi-permeable membrane, your osmosis full definition will go wrong. So there are few keywords in the definition. You have to understand that is the movement of water molecules from their region of higher concentration to their region of lower concentration, or with a higher solute concentration through a semi-permeable membrane. So in this way osmosis is defined. We'll come across an experiment to show how a thistle funnel experiment I have drawn here. You can see here on the board is a lot of experimental questions are set on this experiment. We'll come across it. Please see the board. So you can see here, this is a thistle funnel which is inserted inside a beaker and full supported on a stand, and this thistle funnel is held by a structure called clamp stand, and this is a beaker containing water. Thistle funnel has sugar solution and is dipped into the water solution. That is water, only water, not solution. It's a water means water solution, no other constituent. Separated by a structure, you can see here the red mark I have done, that is cellophane paper. Now cellophane paper is acting here as a semi-permeable membrane. Now it will be left for some time. In this way the arrangement of the experiment was done, where water is taken in a beaker, thistle funnel consists of sugar solution, and separated by a semi-permeable membrane, here it is artificial cellophane paper. After some time it will be observed that the marked initial level of the solution, sugar solution, will rise to the next level, which is written here final level. Now how it has come across, what will be the conclusion here, what was the observation? So definitely here we can understand that water from the beaker through the cellophane paper has entered the thistle funnel. Ultimately that has increased the level of solution in the thistlephenol from the initial level to the final level. So, but not the vice versa, because the more amount of water, so osmosis is a process where more amount of water, if it is present, the region where it is more, it will move from that region to the region where it is less. So that is what osmosis is known as. So here the movement of water molecules was from the beaker into the thistlephenol, increasing the level of sugar solution into the thistlephenol through the cellophane paper, which acts here as a semi-permeable membrane. So we have to understand that in this way, experiment on osmosis, there are different experiment taking animal bladder, which acts as a semi-permeable membrane, viscine bag is there, muslin cloth is there. You could be asked in an examination, what is semi-permeable membrane? So the membrane which allows only solvent molecule, mainly water, to pass from one solution to the another one, or from one region to the another one, is called semi-permeable membrane. They could also ask you, what are the different living and artificial semi-permeable membrane? So artificial semi-permeable membrane, one we got from here, that is cellophane paper and viscine bag, muslin cloth, they all acts as a semi-permeable membrane, whereas animal bladder, like cell membrane and all, they acts as a living cell semi-permeable membrane. So in this way, the root hair cell also takes the water from the soil into the root hair cell, that into the cell sap or the vacuole of the root hair cell through the semi-permeable membrane. They are acting as is cell membrane of the root hair cell. So the water is always passed from the soil into the root hair cell. So this is what osmosis, where the movement is always from the region of the water molecule where it is higher in concentration to the region where it is lower. And osmosis is divided into two categories, that is exosmosis and endosmosis. So exosmosis means exit of water molecule. When it moves out from a cell, that is called exosmosis. There are different examples, like when a cell is dipped into a higher concentrated solution, salt solution and all, the cell loses water by the process of exosmosis, that is exit movement, which is out of the cell. When water molecule leaves the cell, that is called exosmosis. And when it is entry of the water molecule into the cell, if a cell is placed in a solution which is hypotonic, that is more water inside it, then what happened, the cell start absorbing water from the solution. In this way, endosmosis takes place. In my coming video, I will be talking about exosmosis, endosmosis and the tonicity, where you will be cleared about the process of exosmosis, endosmosis and the examples of the iso, hypo and hypertonic, because this is very important in applicative kind of questions that can come. What is hypertonic solution? What is hypotonic solution? What is isotonic? That means mainly they talk about the relative concentration of the solution, and if a cell is placed in hypotonic, what will be the next consequence? If a cell is placed in hypertonic, what will be the consequence? In this way, the examples are set on the basis of the tonicity of a solution and the movement of the water molecule, whether it is inward movement of the water molecule or it is outward movement of the water molecule. Remember, whichever definition of osmosis you are writing, you have to mention about semi-permeable membrane. If semi-permeable membrane is missing in the definition, whether it is osmosis, exosmosis or endosmosis, your answer will go wrong. So this is the most important experiment of thistlephenol. Here two solution could be either water or sugar solution. It could be any other form of solute solution, not only sugar solution. In place of water also there could be any other solution which is less concentrated solution than the solution present in thistlephenol. So they can take here 10% sugar solution, and in beaker they could take 5% sugar solution. In that case also the movement is always from the region where it is more dilute to the region where it is less dilute. So the main, actually the main fundamental behind osmosis is that only, that the movement of water is always from the region where it is more to the region where it is less, when they are separated by a semi-permeable membrane. So this was a short video on osmosis. Stay tuned. Thank you for watching it. Please do like, share and subscribe my channel.